Hi, good afternoon. My name is Danny Shlomo, and today's date is 5/2022/2021. And today I wanted to talk about the regulation of the gastric phase of digestion. So to start off, there are three different phases of digestion. We have the cephalic phase, where once our body senses food, we're able, whether it's touch, sight, smell, through long vagal reflexes, we're able to go to the target cell, which is able to make secretions, as we're going to see later on what those secretions are. On the left side of this figure, once food enters the stomach, we're also able to have the same initial reaction of the target cell and secretions, but this time it's through a short reflex instead of a long reflex as opposed in the cephalic system. Lastly, we have the intestinal phase where food after it leaves the stomach enters the duodenum through the pylori sphincter. It's able to initiate the intestinal phase, as we're going to see later on when we also go over the anatomy of the stomach. So the stomach serves three basic functions, which are storage. The stomach is able to store food for it to be digested and absorbed throughout the digestive tract. The stomach is able to digest food, whether it's mechanically or chemically, through peristaltic waves onto a uniform substance called chyme. The stomach is also able to act as a defense due to the high mucus layer and the low acidity that it has, being able to kill any pathogens that we have ingested along with our food. So on the anatomy on the stomach, if we look at the right side of this figure, firstly we have the esophagus, then we have the esophageal sphincter and where food enters, and then when food enters we have something called receptive relaxation, where the stomach is able to expand and be able to hold and digest the food. So the upper half is not being digested while the bottom half is being digested. It's being digested through peristaltic waves, where it's able to move the food, mix it around mechanically and chemically, and then it's able, once it's digested, it's able to slowly filter out through the pylori sphincter over here to the duodenum, which would initiate the intestinal phase like we were talking about before. Now, how does digestion take place in the stomach? The lumen of the stomach is lined with mucus-producing epithelium that have gastric pits. In these gastric pits, we have gastric glands in the mucus layer, and they're able to secrete various substances, whether it's enzymes in the form of hydrochloric acid, whether it's hormones or paracrine molecules. As we see on this figure over here, we have the G cells able to secrete gastrin, D cells, somatocin, which is considered a negative feedback loop, as we're going to see later on. Chief cells are able to release pepsogen, and once it's activated by protons, it comes pepsin. We also have parietal cells that are able to secrete gastric acid HCl cells. Next slide, I want to talk about it more in depth of how this come to be. So firstly, in this figure, we have the right side of this graph is the cephalic system, and on the left side we have the gastric phase. So both of them, once they're first stimulated, they're able to stimulate the G cells to secrete gastrin, but they have different ways of doing this. So starting off on the right side, we have the cephalic. It's initiated by the long reflex, initiates the G cells to secrete gastrin, while the gastric phase, so that's for the cephalic phase, while for the gastric phase, through food, we have particular amino acid. It's able to stimulate it, and then it's able to stimulate the G cells to also release gastrin. So realize that both of them note that they both have the same end result of having G cells secreting gastrin. After gastrin is released, that would be able to stimulate the parietal cells to secrete protons. Now this would be either directly or indirectly, whether it could be through the ECL cells, which is Etrokofman-like cells, which is able to release histamine, which would then act on the parietal cells to release protons, or directly just the gastrin will be able to secrete, initiate the parietal cells to secrete protons. Now, once the protons are secreted, then it would go through the enteric sensory neuron, which would then stimulate the chief cells. Now, the chief cells would release pepsogen. Now, pepsogen would not be activated into pepsin unless there's protons there. But remember, we had protons that was already released from the parietal cells, so it would right away be activated. Now, we also have a strong negative feedback loop that once there is too much protons or once there's too much pepsin in the system, the D cells would release somatocin, which would be able to inhibit whether it's G cells, to inhibit it secreting gastrin, whether it's the ECL cells to stop histamine, to stop the parietal cells indirectly, or directly stop the parietal cells, or it's also able to stop the chief cells from releasing pepsogen or the activated pepsin after it gets activated by the high intake of protons in our system. So how does the stomach cell have a low pH, right? How is there low pH? So we have mainly a pump where the protons are pumped into the stomach. On the left side of this figure is the stomach, the right side is the intestinal fluid. So protons are pumped into the stomach in exchange for potassium going outside the stomach. Also, due to the protons being pumped inside, the concentration gradient, chlorine would enter the stomach also, and that's how we would get HCl, hydrochloric acid, lowering the pH of the stomach. We're also able to make HCO3 minus, which is from, as we see here, the water is able to combine with protons or hydroxide, and then it's able, with CO2, it's able to make HCO3 minus, and that would act as a buffer in the system, having a higher pH, alkalizing the system. So the stomach also has a defense. It's able to defend itself by the mucus layer, so it has a high mucus layer. Also, it's able to have the HCO3 minus, the bicarbonate that we just mentioned, how it came from CO2 and water, hydroxide and protons, and that is also able to have a higher pH. As we see here, the mucus surfaces have a higher pH, while the stomach lumen has a lower pH, and that's able to regulate the stomach. And it also stops the stomach from odor digestion, from the stomach digesting itself inside. Here is my citations that I used. So just as a brief recap, I wanted to remind you that we went over the 3 phases of digestion, which was the cephalic, the gastric, and intestinal phase. We went over the anatomy. We went over the different secretions and how they are secreted, initiating or starting by the cephalic and the gastric phase through the G cells, gastrin. We also talked about how the pH is lowered in the stomach by the lumen, by the protons in the pump and the potassium, and how Cl enters it after to make HCl. And we also talked about bicarbonate in the mucus layers is able to regulate the stomach, and also highering the pH, and also the mucus is able to protect the stomach itself. Thank you so much.